Hey guys, welcome back to my YouTube channel. So good evening to all, and today I welcome you all for the Study With Me series number 4, and currently we are discussing questions asked in previous year question paper so that we can target DBT BET JRF question paper. Since in this question paper there are many questions repeated, so we'll study the previous year question so that you will get to know idea what type of questions are being asked in the DBT BET JRF examination, friends. So with no delay, let's get started. Amino acid sequence of novel membrane protein has 4 immunoglobulin-like domain and 6 fibronectin-like domain. This protein is most likely it is a cell adhesion molecule. So, cell adhesion molecule will have a 4 immunoglobulin-like domain and 6 fibronectin-like repeat. Okay, and sometimes they will ask what is the structure of cell adhesion molecule, and you should know it has 4 immunoglobulin-like domain and 6 fibronectin-like domain. So the following virus is not transmitted by fecal oral route. So fecal oral route means what? When a disease spreads through fecal oral route, it means that contaminated fecus from an infected person are somehow ingested by the other person. And among this, hepatitis D virus is the only virus which doesn't pass through this fecal oral route. Earliest marker of B cell lineage which appears during maturation of precursor B cells and remain throughout the lifespan of B cells is your B220, which is otherwise called your CD45, and it is the earliest marker of B cell lineage. Which of the following repair mechanism is absent in the cell arrested in cell cycle? Ultimately, DNA synthesizing annealing repair mechanism is deficient in those cells which are arrested in the cell cycle. Next is the intracellular transporting mammalian cell through this vesicular fusion is regulated by which among the following GT bases? So Rho is a protein which is involved in your termination. RAN is also called as GTB binding nuclear protein, and in human it is encoded by RAN gene and whose size is 25 kilodalton, and it is involved in the transport into and out of the cell nucleus during the process of interphase, and it is also involved in mitosis. But they are asking which is involved in vesicular fusion. Next option is RAP. RAP protein or monomeric small guanine triphosphatase. So sometimes they will ask whether RAP is a monomeric or tetrameric. It is a monomeric, and it is function as a key regulator of eukaryotic membrane trafficking. Okay, so the correct option is RAP. Okay, and it is also implicated in budding, transport, docking, and fusion of lipid bilayer vesicle. And your RAS is also involved in binary switches and involved in your signal transduction. HSP17 chaperones are not present in which of the following organ? So they are asking which of the following organ this HSP chaperone are not present. Correct option is your Golgi bodies. Okay, where is your endoplasmic reticulum, nucleus, and mitochondria? The HSP is having. Okay, so it is involved in protein folding also, friends. Major gluconeogenesis occurs in liver and kidney. So whenever you read a textbook in Leninger, first line of this pathway is that it occurs majorly in liver and kidney. Change in which of the following gene is responsible for Fragile X syndrome? It is your FMR1. Molecular basis of this Higashi syndrome is it is due to a mutation involved in regulation of intracellular trafficking. Type 3 hypersensitivity is mediated by immune complex deposition. So if you study this one tabular column present in QB book, it is saying type 3 immune complex mediated. So these things will help you, friends. So already I had uploaded, you refer those videos. A child stung by a bee experiences a respiratory distress within minute and lapses into unconscious. The reaction is probably mediated. So it is your IgE antibody, and it is a type 1 hypersensitivity reaction. Neutrophils are attracted at your infected area by phagocytosis of your IgE coated bacteria. Chromatin loops are formed by periodic attachment of falling onto nucleosylation. It is your mass. It is not a it is matrix attachment region, and it provides a scaffolding protein. Telomeric region doesn't contain AT-rich sequences. It has G-rich sequences, quadral looplets, and it forms a TND loop. colon modification of histone that is not known to play a role in regulation of gene expression. The correct option is your glycosylation. Okay, whereas other colon modification of histones like poly ADP ribosyl acetylation methylation are being involved. Ogasaki fragment or what? It is a short structures or short stretches of DNA which attaches to RNA primer on a lagging strand. So this is the Ogasaki filament in logging strand. It forms a small segment and the segment is called Ogasaki filament. The changes in electric potential for neuron that constitute action potential of following order. So first is the resting potential, next is depolarization, followed by hyperpolarization and your resting potential. This is the order of your action potential. Suppose you are treated a normal mammalian cell with a substance, it will inhibit the NK-ATPase pump. Already we saw what is mean by NK-ATPase pump. It will transfer 3 sodium ions out and it will import 2 potassium ion in. And the major role is that it is involved in maintaining the membrane potential of cell. So when we inhibit this pump, then ultimately your cell membrane potential would become zero. It won't be in a normal range. Developing T cell that reacts strongly with a cell peptide bound to a self-efficacy molecule, or it will eliminate in the thymus where your anti-differentiation of your T cells will occur. Which of the following statement is correct? These lymphocytes are conditioned by the bone marrow. Yes, it is a correct option because B lymphocyte, the maturation and the differentiation will occur in bone marrow. But here they are saying thymus. Okay. B cell produce, sorry friends, the correct option is your B cell produce plasma cells and memory cells. Whereas T lymphocytes are conditioned in thymus and B lymphocytes are conditioned by thymus. No, it is conditioned by bone marrow. T cell will produce cytokine, but here they are saying T cell don't produce cytokine. Which of the following force among the following is primarily responsible for stabilizing the tertiary structure of globular protein? So which is a correct option, which is involved, and the correct option is your hydrophobic effect. It is involved in stabilizing the tertiary structure of globular protein. Which of the following sequence describes the passage of action potential to the neuron? The correct option is your dendrite, cell body, axon, synaptic cleft. It is a correct option, friends. So if you see, first dendrite will occur, it will receive the signal, then it will go to the cell body, then axon, then your synaptic cleft. So this is how your thing will pass. First dendrite, then your cell body, then axon, then last synaptic terminal. Some extremozymes are found remain active and stable up to 140 degrees Celsius. The decreased flexibility and rigidity of the molecule is due to a reduction in the glycine content. Extremophiles will found remain active and stable up to 140 degree, and this rigidity is due to the reduction in the glycine content of the protein. A silent substitution means, okay, it will substitute one amino acid for a different amino acid with a similar property and it won't affect the protein function. Okay, so it is the correct option for your silent substitution. The next option is that sorry friends, a silent substitution change one DNA base in the codon without changing the amino acid sequence. So it will silently do all kind of activity. Satellite DNA are not typically found within which of the following pact of chromosome? Silent DNA are not found in your heterochromatin region, and heterochromatin is the dense material, and this heterochromatin is generally found in centromeres and telomeres. Satellite DNA is not present in your heterochromatin region because they are not transcriptionally active. Which are the following sugar to sugar linkage among the following in 5' molecule? It is your 5' molecule. Introduction of nonsense mutation in eukaryotic protein gene often lead to increased degradation of, what is nonsense mutation? It will lead to increased degradation of mRNA in the cytoplasm. Next thing is that what is the effect of the N mutation having a phage lambda infection? If any of the phage is having N mutation, then it will completely abolishment of infection. So there is no lytic cycle or lysogenic cycle possible. Bacterial FTS gene is required for the septum formation. Two plasmid are of same compatibility group if they cannot coexist in the same bacterial cell. They cannot coexist in the same bacterial cell. It is the correct option. So the next thing is that Alu elements are example of the lines, long interspace nuclear elements. Telomere consists of simple repeats of your CA-rich strands that will interact with the protein. So telomeres are having the CA now, CA bond. 6 molar guanidine hydrochloride is known to denature. So this guanidine hydrochloride is used as a denaturing agent, and also 6 molar is efficient. Such a high concentration is able to break down the non-covalent forces sustaining the structure of protein by affecting the electrostatic, hydrophobic and hydrogen bond interaction. The next thing, which of the following amino acid is critical for a folding of protein due to slow isomerization of peptide bond preceding this amino acid depend upon solvent environment is your proline. Proline is amino acid which is involved in folding of protein by slow isomerization of your peptide bond. In peptide values around dihydrogen angle. So you should study about this pi angle and your di angle, friends. It will be definitely asked, and pi angle is the rotation about n c alpha bond. Next thing is the each of the following technique cannot be used to determine the molecular weight of protein. It is your UV absorption. It is not able to determine the molecular weight of protein. Beta globulin, which is a monomer at neutral pH, is known to tetramerize at an acidic pH of 2. Each of the following technique could be effectively employed to demonstrate the formation of tetramer. The correct option is your native gel electrophoresis, and in native gel electrophoresis, protein will be separated based on your net charge, size, shape of the native structure. Which of the following is incorrect? So the incorrect option is what? Lysosome synthesize molecule for extracellular protein degradation. So we are asking among which is the wrong option. Wrong option is your lysosome, which is involved in degradation, whereas your mitochondria is involved in cellular respiration. Endoplasmic reticulum, it synthesizes a protein and send them to Golgi apparatus, whereas polysome, it is involved in making a large quantity of particular protein. Which of the following statement is true about heritability is true, asking, and it is a measure of proportion of variation which is contributed only by genetic matter. There is no environmental factor being acting upon them. Next question is the state is one, okay, which is present in few discrete classes, but it is influenced by environmental factor. Each of the following describes a polymorphism that occurs within a gene that causes a Huntington disease. Huntington disease is caused by short tandem repeat polymorphism. 5-bromouracil, what does the 5-bromouracil do? 5-bromouracil replaces the G. What is the role of 5-bromouracil? It will replace the T and it will bind to G rather than that of A. So we will deal them separately, friends, don't worry. A hemiotic mutation is one in which it substitutes one body part for another during the process of development. Each of the following causes deviation of Hardy-Weinberg law is ultimately a small population size. Whereas Hardy-Weinberg law, they say large population size, there should be random matting and there should not be any selection pressure and there should be no mutation or migration. Okay, cell division cycle is divided into 4 phases, G1, S, G2, M. Standard eukaryotic cycle are 12 hours or longer duration. Early embryonic cell cycle are extremely rapid, having a duration of less than a hour. So they are asking which of the following phases are drastically reduced in the embryonic cell cycle. So here G1 phase and G2 phase are two phases which will drastically reduced in the cell cycle. So that's it for today's video, friends. We'll continue our question paper studying in our upcoming videos also. So follow my channel and we'll definitely crack the DBT BET JRF examination. Thanks for watching this video, friends.